Hi everybody, we are here at Imperial College and this is the very start of the Smart Cells project, and we're going to take you on a tour inside this laboratory here, and we're going to meet the incredible scientists who are trying to make life better for patients with myeloma, and we're going to find out what myeloma is. Everybody ready to go in?
Yep.
Excited? Have you been in a lab before?
No.
Once.
I
had to get to my lab though.
Hi. Just before we go inside the lab, we all wanted to show you some of the slides that these amazing scientists have sent us about what they're doing. They sent us a slide explaining that they're actually working with cells that are part of the human immune system. They showed us loads of white blood cells, and one in particular was this special smart cell that they're researching, and that was a T cell. So if you're looking at this diagram, check out the green T cell with a antigen receptor sticking on the side. These T cells are really important and they're crucial for long-term protection against diseases. Then they explained that they weren't even just using regular T cells. They were using iNKT cells, which are a subgroup of T cells that can naturally recognise and rapidly kill cancer cells. And these T cells also have a minimal risk of rejection, which is pretty useful. And then they explained that instead of just using nature, they soup it up with their amazing genetic engineering, and they add an extra antigen receptor, a chimeric antigen receptor. So it's like the cell's got two weapons, double the amount of weapons, and it basically means that it can recognise a really much wider range of cancers. And also, those type of cells can be prepared in advance and then given readily to the patients. So the final diagram, they showed us their process. So you can see blood vial on the left. So they take the blood from healthy people, they modify it, they add in the extra receptors. So now you've got CAR iNKT cells. They grow loads of them in the lab, rapidly expand the number. Then they freeze them and store them. And then they give them to patients in an infusion. And finally, we see the cell exploding the cancer cell. But before we say any more, let's go in the lab, follow us, and we'll go and see what's going on.
iNKT T cells, natural killer T cells.
Wow.
That's one of the subtype is I, is the invariant TCR we have. So there is a subtype of iNKT cell. So this iNKT cells can do many jobs. So I can take you to the welcome to our immuno lab.
How does an NK T cell kill a cancer cell? So it's not like in some, we learned before that some of the immune cells kind of reach around a cell and dissolve it and eat it. But how does that cell kill a cancer cell? How does it do it?
Okay. As I said, when we have something like infection, there are two ways, right? So one way is random killing. So basically they chop it and eat it, like macrophages can come and engulf. Basically they are there, they can come and eat it, right? But these iNKT cells, they cannot eat it like them. So what they will do, two ways they will act. One, if they find the target, they will go and attack the cells and make a pore into the cells and release some kind of granules like perforin granzyme B. They can just introduce this into the cells and this will be toxic.
Like a kind of weapon to attack it back.
Yeah, cell warriors.
Holes and the weapons go in, and then boom, the cancer cells exploded and go.
Yeah.
And exactly, I think what you said, exploded, is the right word as well, because then the cancer cells, in a way, make bubbles. So then they are completely destroyed. So they just don't disappear, but they make bubbles, apoptotic bubbles in a way. And then they're just in very small pieces like debris, and then they're dissolved.
These Hana, BSc student and the class T researchers study. So they are all working on immunotherapy. They are all they all dealing with cancer cells. They are dealing with iron cells. So our ultimate goal is to understand these iNKT cells. We know how they work based on our data, our understanding in our lab, and also others from all over the world.
Something really interesting on the question that you asked before, just to understand the differences between CAR T cells and CAR NK cells, is that, for example, for CAR T cells, each patient with cancer has to provide their own T cells, and then they are manufactured and engineered in a way that they treat their own self as well. But for iNKT cells, as Raju said, we can select some selected donors, and then we can engineer these cells and provide it to different cancer patients. So you don't have to have specific T cells from each patient for their own cancer, but they can be distributed around patients. And I think that's very important for iNKT cells.
So, we already have
iNKT.
we all already have them.
Yeah.
So the treatment is manipulating those cells that we already had. Is that right?
Yeah, that's right.
Okay.
It doesn't mean that it has to be from the same individual who is suffering already, but it can be from any individual.
So once you have adapted the iNKT cell, is that got a new name when you put them back in?
Well, we say when we take them as the iNKT, but when we give them back, we say CAR iNKT. But CAR.
So what are you seeing in there?
So in here, we have some very small cells, circular cells, which we call lymphocytes, and in particular they are the iNKT cells, so which is the subset that we are trying to do the research on. And by observing them under the microscope, you can see how they proliferate, if they are healthy, if they have been contaminated, how they are growing, if they interact with one another, and depending on the type of cells, we can see their morphology as well. So these are the main things that we're trying to see when we investigate them under the microscope.
Here you can see if the cells are growing, you know how they make them friends? I'll show you. Do you see clumps?
Yeah.
Yep.
You see how they are clumping together?
Yeah.
How they are going to be friends.
Yes.
You see? They have this capability.
What's happening in the club?
So they are iNKT cells. So they can communicate each other. When they are communicating each other, let's say if you put one cell here and one cell there, they won't be able to communicate. Right? If you put them next to each other, they are like friends.
Oh, like they're weird. Closer together, we can pass the message on a bit better than if we're like on the other side of the car.
They're very happy. They'll grow faster.
I'm looking at just smart cells, iNKT cells, and how they can be used to treat myeloma. And myeloma is a type of cancer in the blood. And what I'm doing in my project, I'm trying to combine some drugs with the iNKT cells to see if we can really enhance the killing of these cancer cells by the iNKT cells by adding drug before. So in my work, I'll get my cancer cells, I'll add them to my plate, and then what I'll do is I'll add my drug. So the drug I'm working with is called Atra, and this Atra should help my cancer cells express something called CD1D, but it's basically just something that should help the iNKT cells to bind to the cancer cells and kill them. So add the drug first, leave it for a certain amount of time. This could be 1 to 2 hours, it could be overnight, and that's one of the things we're testing. And then what I'll do is the next day I'll kind of spin the cells and remove the drug, and then I'll add the iNKT cells and leave it for some time. And then I'll test, okay, how much death do I get with this drug and this iNKT cells in comparison to just having these special smart cells. And that's what I'm comparing and seeing how effective this drug is at boosting iNKT cells and their effect on the cancer cells.
That's so exciting. Just thinking that you can boost up these amazing smart cells to make them even more effective is brilliant. So if a student at school perhaps is watching this video, they might be learning about variables and things like that. So the variable bit, you're sort of using smaller or bigger or different quantities of that drug, and seeing what happens. And hopefully, when you come back to the lab, you find out good results. Like you might find the optimal amount of the drug to maximise the smart cells to work. That sounds exciting. Is it all in liquid form, and do you kind of squeeze it in with a pipette or?
So my drug will come prepared as liquid form, then I'll dilute it with something. So I guess it's like a vehicle to dilute it. So then can have the different concentrations, and then I'll get the pipettes and I'll just put the drug in and make sure it's well mixed. So yes, I'm kind of making dilutions with my drug.
Is it exciting working in a lab? Does it surprise you sometimes when you come back and you find the results? Do you ever think, oh wow, that's exciting? Or
yes.
How does it feel?
I think the most surprising thing is that before this I was in like hospital doing like medicine, and now I'm working with like cells. So you go from working with people to working with cells, and it's like you're very delicate, very precise, but also with people you have to be quite delicate, and so it's all about, I guess, applying what I've known in medicine with people, so like cells, and working with cells and looking after them. They require lots of care, and you really have to like look under the microscope every day and take care of them. So it's really interesting to kind of work with something so small but needs so much care and attention. And I guess that's been the most fun part. And I guess it's also fun because, like in everyday life, your science can be unpredictable. You never know what you'll find. So, you might go to your results and it's what you expected, or it might be even better, or maybe not something, maybe you're not too sure. But that's all that science. You kind of be working back and you're looking at what you've done and improving what you've done. So yeah.